Dear students, I am going to discuss about the Newcastle disease of poultry. This disease is caused by the Ebola virus belongs to the family Paramyxoviridae. This virus is RNA virus and it is enveloped having herringbone shaped helical symmetry. It is 150 to 350 nm in size and this virus having non-segmented negative sense single stranded RNA which is 13 to 19 kb in size. This virus has two types of glycoproteins or which is project from the envelopes, attachment proteins and fusion proteins. Attachment protein contains hemagglutinin neuraminidase protein and fusion protein which is responsible for the fusion of the envelope to the host cell membrane. The another protein also present which is associated with the matrix of virus known as matrix protein. This disease is also known as Ranicate disease, pseudo-fowl pest, avian distemper, avian pneumoencephalitis and pseudo-poultry plague. This Ebola virus is having 9 serotypes of the Newcastle disease virus or avian paramyxovirus. The avian paramyxovirus 1 is the main causal agent of the Newcastle disease virus. The main host of the disease is poultry, duck, geese, but parrot, ostrich and migratory waterfowl can also infect. This virus replicate into the cytoplasm so that intracytoplasmic inclusion bodies are formed. This disease was first reported into the Newcastle place in England. That's why it is known as Newcastle disease. And in India, it is first reported into the Ranikhet area of the Nainital region. The virus has the property of the hemagglutination of the mammalian and the avian RBCs. The virus can be easily inactivated at the temperature of 56 degrees centigrade in 3 hours and at 60 degrees Celsius for 13 minutes. The mortality rate of the disease is 90 to 100%. It depends on the virulence of the strain and vaccination status of the poultry. The virus can be transmitted through the any route like inhalation, ingestion or direct contact. The virus cannot be transmitted through the vertical transmission, but chicks can be infected in hatchery via contaminated eggshells. About pathogenesis of the virus is first initially replicated into the mucosal epithelium of the upper respiratory tract or intestinal tract because it is a epitheliotropic virus. After infection, virus spread via the blood to the spleen and bone marrow and producing a secondary viremia, leading to infection of other target organs like lung, intestine and central nervous system. Respiratory distress and dyspnea result from the congestion of the lungs and damage to the respiratory center in the brain. The gross pathologic findings includes hemorrhages in the larynx, trachea, esophagus and throughout the intestine. There are many pathotypes of the Newcastle disease virus: viscerotropic velogenic strain, neurotropic velogenic strain. The velogenic strains are the highly fatal strain which cause the intestinal lesion and the nervous or respiratory lesion respectively. The mesogenic strains are less pathogenic than the velogenic strain and which cause the respiratory signs or affect the respiratory systems and occasionally cause the nervous signs but low mortality. The lantogenic strain is causes mild or subclinical respiratory infection. Different strains are responsible for the different forms of the disease like dolls or asiatic swarms caused by the velogenic viscerotropic strains, and chickens are mainly affected through this strain, and in this form the respiratory and intestinal tract affected and hemorrhagic lesions are seen, which is form or pneumoencephalitis form which is caused by the velogenic neurotropic strain and mostly neurological signs occur. Boarded form, and this form is caused by the mesogenic strain and respiratory signs occur occasionally, nervous signs can be seen. Hitcher form, which is caused by the lantogenic form, and mild respiratory symptoms are observed in this form. The incubation period of the disease is 2 to 15 days, and in respiratory signs, dyspnea, gasping, nasal discharge, and cyanosis of comb and wattles are observed. In intestinal form, crop will be dilated and fibrinous exudate in pharynx, yellowish greenish diarrhea, and such type of lesions are present in the intestine. In nervous signs, drooping of neck, twisting of neck, paralysis of veins, and nerve of nerves occurs. Edema of head and neck also observed, and in the pathognomic lesion, typical pinpoint hemorrhages in proventriculus and gizzard are seen. For the diagnosis of the disease, the sample should be collected from the trachea, cloacal swab, nasal secretion and feces. From the dead birds, oral nasal swabs, lung tissue, kidney, spleen, brain, liver and heart tissue can be collected. For the isolation of the virus, embryonated chicken egg, which is 9 to 12 days old, are take are used, and sample first sample was processed, virus inoculum prepared, and thus this inoculum is inoculated into this embryonated chicken egg through allantoic sac route, and after 24 to 72 hours hemorrhages and encephalitis was observed. Uh, the allantoic fluid is further tested for the hemagglutination activity, and hemagglutination inhibition test using specific antiserum confirms the presence of Newcastle disease virus. The another diagnostic test like immunofluorescent test, ELISA and RT-PCR are also used. Virus can also be grown into the chicken embryo fibroblast culture and in BHK21 cell line. For prevention and control, many hygienic measures should be taken, like the location of poultry farms should be apart from the residential area. Thorough cleaning and disinfection of vehicles and equipment should be occur. Movement should be restricted in between farms, and isolation of the diseased birds. And vaccination. For the vaccination of the disease, many live virus vaccines are developed using different strains like strain F, strain B1, Lasota strain and CDF66 strain, and these are the lentogenic strains. Mesogenic strain of NDB are also used for the vaccine preparation, such as Micteshwar strain, R2B strain, Kamurev strain and Ryukin strain. The vaccination should be done through the spray or drinking water or by intranasal route. As per the vaccination schedule, the first vaccination should be done in the first week by using F strain or HRB1 strain, and at the fourth week of the age, the second vaccination should be done via oral vaccination in drinking water with B1 strain, and at the 6 to 8 weeks, the intramuscular route and Vib method route are used to introduce the Micteshwar R2B strain for booster vaccination. This is all about the Newcastle disease of the poultry. Thank you.